get started then. Okay, so I'm going to be presenting how to use DeCapo, how to employ established deep learning techniques without coding using DeCapo. So DeCapo is a framework I'm working on to help run sort of common machine learning jobs that I've done at Janelia through a web front-end.
Hey, Will, just real quick, your microphone volume is very low compared to everyone else I've heard.
Let's see if I can fix that. Is this better?
Yes, thanks.
Perfect. All right, yeah, so DeCapo employ sort of established techniques that machine learning techniques that are used at Janelia quickly and easily through a web front end. And so this hasn't just been my work. I've gotten lots of help from David and great advice from Stefan, Jan, Ben, and Adam. So I always like to start with a little cartoon to help motivate DeCapo and familiarize people with the components. So the way I think about it is you've got some machine learning project and you've got someone who's going to be working on it and he's excited to get started. So the first thing that you do is you generally collect some data and you want some results, and then the machine learning is the step in the middle that you have to do. And so I'm assuming the data has already been collected and you're now focusing on this machine learning part and looking at the results. So this isn't too bad, but then quite often you want to apply a couple different techniques. You want to search across parameter space. Maybe there are different models you want to try, different learning rates. Essentially, in machine learning, a large part of the work you do is just searching over the parameter space. And so this can be a bit repetitive. It can be a lot of coding to code little little scripts to parse different parameters that you want to use, but it's not too bad. Where it might get a little daunting is then when you've got many data sets, all of which you want to analyze with many different parameters, and then you have a lot of results to analyze at the end. So our goal with DeCapo is to basically factor out this middle part and minimize the amount of code that you have to duplicate and just minimize effort here in the middle by using common parts of the machine, basically plugging together common pieces that we use for machine learning here at Janelia. And a lot of this can be done because the volumes and data sets we're working with here are typically very similar. So they're going to be large microscopy volumes, light microscopy, EM microscopy, stuff like that. So then I like to emphasize here that DeCapo is a tool that helps you do your machine learning. It is not all, like, it won't do everything for you, right? So if you want to browse the web, you might use Chrome. There are other tools, but hopefully DeCapo will help you do your machine learning easier and faster. So looking at DeCapo as a whole, we have split the machine learning job up into 4 main components that you can configure individually and then ideally plug together to get lots of different jobs up and running. So the first thing you're going to do is you're going to need to split up your data. And so we call this the data split. And this is how you're going to separate your data between training and validation. And so hopefully this is familiar to anyone with a machine learning background, but you always need to keep some amount of your data held out. Your model never sees it. And so you can evaluate how well you are generalizing to data that your model hasn't seen before. If you don't do this, you always run the risk that your model just essentially memorizes your training data and outputs very great results, but then when you try to apply it anywhere else, you just get sort of nonsense outputs. So this is the first main important step. You need to make sure you've got a good training set and a good validation set. The second step is to choose your architecture. And so commonly we like to use the UNet. This has been applied for many different tasks very successfully and works very well for essentially whenever you want an output of a similar shape to your input. So you input a volume, you get a volume out. The task is a little more complicated, but this is essentially mapping from the, it provides a couple mappings. So one thing you want to do is you want to transform the ground truth into a target. The target is what you're actually going to learn with your model. So in this case, I'm showing an example of a signed distance transform. So your input is a binary mask, and then you do a distance transform. And so the further inside the object you are, the more positive, i.e. green, your target is, and the further outside of an object, the more red or negative your distance is. And we pass this through a tan-H activation. So the distances plateau at ±1. So now once you can map from the ground truth to the target is what your model is going to predict. And your prediction will hopefully look very similar to your target. At least through training, you hope to bring them closer together. But then you also need to map from the prediction back to something that looks like the original ground truth, because typically you want to provide some ground truth in the format that you'd like, and then you want your model to output the same thing on new data. So not only does the task define these transformations from ground truth to target and prediction to output, but the task also needs to define how to compute accuracy. So on the from the target to the prediction, we need a way to calculate the accuracy in a way that is easily that gives you an easy gradient that you can backpropagate through your model. So this is sort of the standard part of machine learning that you might hear about, the loss. So you need to be able to calculate a loss on the target and prediction, which then gets backpropagated and upgrades the weights of your model according to the gradient. And this is how your network gets better and better at producing the prediction from the input. And then, so that will tell you, the loss will tell you how to update your model given a target prediction. But we also want to know exactly how well the model is doing on the task that you care about, which is essentially comparing the ground truth to the output. You could get arbitrarily good at, you could get arbitrarily low values of the loss. But depending on the task, depending on the loss, and how you transform back to something that looks like the ground truth, that may not translate well to great accuracy. Okay, so that's the task. It's a bit of a more involved component of DeCapo, but I hope that all makes sense. It's these two mappings and how to compare them. Finally, the trainer defines how we want to essentially pass data from the data split to the model using the task. It essentially ties the other three together. So it's got a couple different important components. The main one probably being augmentation. So given some data, like some raw data, some ground truth data, a target, you can commonly apply augments that will randomly perturb your data, which will essentially just increase the amount of training data you have. So machine learning models are famously very data hungry. You need a lot of it. And one way of sort of faking having more is applying augmentations. So the sample I have here on the left is essentially taking this watermelon and just rotating it. Other examples would be adding noise to your raw data. It could be random, it could be like uniform, Gaussian, gamma noise, whatever sort of noise you want. You can also rotate, you can stretch and deform, you can mirror, transpose. These are the augmentations that are commonly done on volumetric data, especially microscopy data, because these are the things that you want to be equivariant over. So if your raw data is mirrored, you want your ground truth data to be mirrored. It's very rare. I guess there are cases where you might not want that, but usually you do. So those options are in. And then the trainer handles sort of the main loops that come with machine learning. So you need to keep fetching these batches, keep selecting random parts of your training data, augment them, actually use the model to predict the result or the target, calculate the loss, run your backpropagation, and then maybe save any snapshots so that you can see what's going on during training. So that's the trainer. He's sort of really combining the other three to actually run your model, run the train. So finally, you don't need to define just one of these. You can have many data sets, many trainers, many architectures, many tasks, and combine them so that you can do this search space over many different parameters. So at this point, I want to jump over to the DeCapo front end and show you how to actually use the tool. So over here on this is the DeCapo dashboard. There's two main tabs to look at: monitor runs and create new runs. So I'm just going to pretend like we have created a run. We've configured all of these components, and we want to see what the output of DeCapo will look like when you're done. So let's take some models I've already trained. We select the configurations that went into creating this model, and here I've got 3 runs that have finished. And so let's plot the losses. And so this is essentially what we get out of DeCapo at the end. We get a nice loss plot. So this is how well you are performing on the target tasks, how close your target is to how close your prediction is to the target. And then for validation down here, I'm plotting the Jaccard score on ECS. So this is extracellular space on a liver data set. The important thing to note here is that loss you want to minimize, this validation score you want to maximize, and we see it going up here, which is ideally what you would like to see. so once you analyzed your results, you see that, okay, we are training decently. You may want to look at your data. So for this specific task, we see that we're doing okay. Let's look at the actual data that went into it. So here there are helpful links for looking at your data splits. This is quite nice just to make sure that your data is loading in properly. You've got your training data set is what you want it to be, your validation data set is what you want it to be. So when you click on it, it'll load in here. So this is a NeuroGlancer viewer that will load all of your avoidance in. And you can click on them here. This is the raw data for the liver that these models were trained on. And then you can load in a couple training crops. And if we look out in the 3D viewer, not sure why they're loading in at the corner there. Well, I loaded in, I preloaded some of this data so that we can go look at here. All right, so on the left are the training crops, and on the right is the validation crop. So we can jump around to a couple of these and look at, so, right, we see the raw data and the validation and the ground truth labels. These, this is work by the awesome annotation team in CellMap, Grace and Allison. They've done a lot of this annotation for us and provided very nice high quality labels here. And so these are a whole bunch of different organelles annotated at 2 nanometer resolution. The raw data is 4 nanometer FIBSIM. Yeah, this is an example of one of the training crops. We can see in here that a whole bunch have been labeled at various different locations. The validation crop over here is a little bit larger, with ideally you want a good spread of all the different organelles that you want to annotate in here, or that you want to predict at the end. So we've got lots of things like the nuclei, the nucleus down here, mitochondria with their membranes, lots of ER sort of going all throughout here. And so these are the kind of things that we want to predict. Yeah. So going back to monitoring, this is what we see. So the loss would be, that's the score that we're getting on the distance transform, how close are our distances to the true distances. And then the validation score here is for a specific organelle, ECS. So if we go back to the validation data set here, ECS is this blue region over here, extracellular space, that is right, sort of entering into this large region there. And so our predictions are getting better there. And so, okay, we've looked at the input data. We also want to look at the output data. So now, once you see that you're performing okay, you want to, or once you see that quantitatively you're doing pretty good, you probably want to get a qualitative understanding of how well your models are performing. So here we provide another helpful link on the run itself that you can open, and this will be pretty busy. So it loads in snapshots that you've made during training and the validation volumes that you predict at fixed intervals. Since this is a lot of arrays, I've also preloaded in a view with this sort of filtered down to something that we might want to look at right now. So let's look at the validation output first. So this is the same queue that we just looked at in the raw data, right? So here is, this is the validation volume input. And now we're looking at the results from the model. So we just see this, this raw crop going in. And then the output is, I visualize mitochondria here because they're nice and easy to look at. So the ground truth mitochondria right here, this is basically just taken the two labels that we saw related to mitochondria down here. There's the mitochondria lumen, and there's the mitochondria membrane. And so these have been combined. Oh, there's also these little mitochondria ribosomes in here. So these three labels have been combined into a single mitochondria channel, which we are predicting as a whole. So this is the ground truth for mitochondria. And if we load in the predictions, the predictions are in blue and the ground truth is in green. So you get sort of a cyan color where they overlap, and then blue where you have false positives. And as a whole, the predictions are starting to look pretty decent here, with a fair number of false positives. So that's the validation output that you can see from the front end. The next thing that you also sometimes like to look at for debugging purposes is the snapshots. So this is what a individual in a single training sample, what the model sees, what it predicts, what it and a couple other things. So first off, there's the raw. Here we see the prediction on top of the raw. So I am I'm visualizing ER here. Yeah, ER and ER membrane here. And so we can compare to the ground truth. What's nice about neurogrants is we can also put them next to each other. So there's the raw and the ground truth on the bottom, and the raw and the prediction on the top. And so the prediction here is just the distance transform that I have. I've emphasized it quite a bit. I've scaled it up quite a bit because for small objects like this, your positive distances are going to be quite small. So if you don't scale it up, it's pretty faint. But that's what our distance predictions look like compared to the ground truth. And a nice feature of the of looking at snapshots is you can also look at gradients. And so these are sometimes quite hard to parse. But if you take some time to sit down and really look at what your gradients are, it can help you get an understanding of what might be going wrong in your model in cases where it's not learning what you want it to learn. So this is a feature that I don't really expect most people to use, but if you're more familiar with machine learning, it's very nice to have. Okay, so I think that's it for the liver and
yeah.
Can I
um, for neuroglancer, here you have, when you were looking at the, yeah, so this doesn't have the characteristically long URL. How do you achieve these little tiny URLs?
Right. So if you load it through NeoGlancer, right? You open this link, you are going to get a very long URL in here. This is massive.
Right.
Comes with everything you need to view it in a browser. Over here, the problem with this is that it's loading from a file server, and I don't have a dedicated file server. I've just spun up a somewhat slow Python script on a small compute node to run the file server. So what I've done over here instead is just load these volumes into Python, and then basically launch the NeoGlancer view straight from my workstation. So I don't have to rely on the file server, which can be a bit slow.
I see. So you have this is a Python server that's serving also the NeoGlancer front end.
Yeah. So this isn't using the publicly hosted NeoGlancer either. This is generating a NeoGlancer server, loading the data directly into it, and visualizing it. I find that to be quite a bit faster and nicer to use for live demonstrations than relying on the publicly hosted NeoGlancer and a file server, which can be slow.
Okay. Yeah, that's
especially for larger volumetric data sets like this, I find it helpful. If it's a smaller data set, then sometimes I'll just go with the file server. But I think that's it for the liver, and gives you a good overview of everything you get out of DeCapo. You get a good view of the input data. You can view the output data in the form of snapshots that you get during training for debugging, and also validation blocks that you can use to qualitatively analyze how well your models are doing. And then you also get these plots, so you can get a quantitative idea of how well your networks are doing. So now I'm going to switch to a toy model, since we still have quite a bit of time left. Toy dataset, toy model, all of that, so that we can actually run. We can look at the configurations, and we can run something now and hopefully see some results soon. So I'm just going to close these links that I made earlier. If I can reach the button behind this, the share screen info. Okay. So to start off with the task, I've already generated configurations for this dummy task, and run some of them in advance. So even if this does take a little bit of time, we should be able to see what would come out after an hour, after a couple minutes or an hour or two. All right, so let's start with the task. So creating a task is not too bad. So we're going to predict distances. We basically just need to give it a name. So we're going to predict the distance to an epithelial cell membrane. So the number of channels that will output, will just output a single channel membrane. So on the liver project that I showed you earlier, we were outputting a whole bunch of different channels. We had mito, we had nuclei, we had ER, all that. This time we're just sticking to single channel membrane. Clip distance and tolerance distance. This is for calculating some validation scores. If you right, so if you have sort of point-like structures, for example, you might want to consider them false positives only if they are above a certain distance from the true object, right? So if you have, right, if you've got a point in your ground truth and your point that was predicted is a couple pixels off, you might still want to consider that a true positive rather than a false positive. So that's what the clip distance and tolerance distance let you do. They let you control that a little bit. And then the scale factor is how much you want to scale your distances by before you apply the tan-H normalization. So tan-H will saturate very quickly. I think around like 3 or 4, it's already close to 1. So if you provide a scale factor, you will you'll divide your true distances by the scale factor before you pass it through the tan-H. So this will help you get like a smoother drop off in distances over space. And then mass distances, this is whether or not you want to sort of mask out windowing artifacts at the boundary of your object where you may not know the ground truth data. So we can save that. And that's the task done at epithelial membrane right there. For data splits, so I'm not going to go through and plug in values for the data split. I've preloaded that in here to show you what this looks like. So this is generally the most painful part, in my opinion, of setting up a new project in DeCapo, is you have to point to all of your data and where it is. So at the moment, the only really supported option is data living on the Janelia file system. So either in DM11 or NRS, both of those work pretty well. And then you generally want to store your data in ZAR or N5 or some other chunked format for machine learning so you can randomly access various bits of it. But getting back to my point, this is quite a bit of configuration that you need to plug in. And a lot of it is providing paths to data sets that live somewhere on the file system and providing what sort of where in that ZAR is your data. So going down from top to bottom, we need a name for our data split. Epithelial, I've already configured it. This is just essentially a cut down version. Then we have our training configs. So this is the data that we're going to use for training. Name, this is the name of one of the samples. Raw config. So now the raw data is going to be a set of intensities. We give that a name, so it's the same name as this data set, but now this is specifically the raw data, which is being pulled from a ZAR array. So here we configure the path to that ZAR, the data set we want to read from, and a name. And then optionally you can provide axes. I think this is going to disappear as we rely more heavily on the actual metadata of the ZAR and require you to provide that data in the metadata. The nice thing about the intensities array is that it lets you now define the minimum and maximum, essentially the range of your data. So if you want to add noise if you want to augment your training data with noise, then it's very helpful to know the range of your data in advance before you apply that noise, so that you can add stuff that is in sort of the same relative scale, right? It won't really make sense to add Gaussian noise or uniform noise between 0 and 1 if your intensities are between 0 and 255. It just won't make much of a difference. So that's why the intensities array lets you provide the range here. So it will handle all that for you on the back end. To go along with the raw config, we also have a ground truth config. And this is where we are going to binarize our array. I think it makes sense to directly open up the already configured version and see what this data looks like. Right, so here I also already have this loaded in as loaded in and cleaned up, right? So when we clicked on the link, we got this very noisy view with a lot of, so I've configured quite a few training volumes in here, which is nice to have for training, but not too nice to have for visualization. So over here, I've cut that down. So this is what our data looks like for the epithelial toy example. We've got our raw data, which is their intensities between 0 and 1. And then we've got our ground truth data, which is essentially just a cell versus membrane segmentation here. And so the membrane is just this 1 pixel thick boundary between all the different cells. And what I want to do here is as like just a sample task is to predict these membranes. So if we zoom in a bit, we can see that the membranes have a label of 0. You can see on the PER02_30 ground truth. The label is 0 here. If you hover over the cell, it's 1. If you hover over the membrane, it's 0. So that is why when we're creating this data set, we want to tell it which array, so where to pull the ground truth data from. We essentially want to invert it, where the membrane is now going to be the class that we want to predict. So the name of the array, the source where the data is going to come from, it's going to come from a ZAR. It's a foreground background. I'm just calling it foreground background there, mostly because the volume that the data is stored in is called ground truth foreground background. And again, I have to pass it the path to the ZAR I want to read from. And then down at the bottom, the groupings. This is the important part of the binarized array. It lets you define a class and then define what labels are part of that class. So we want to look at the membrane, and as we saw in the NeoGlancer link over here, the membrane consists of label 1. Incidentally, this is also how we did the mitochondria in the liver. Remember, we had lumen, membrane, and the little ribosomes in the mitochondria. So here we would have provided labels for all 3, and they would have binarized into a single channel with just a sort of conglomerated mito class. So that's it for a single training crop. We now pointed it to the raw data. We told the raw data what range it was in, and we pointed it to the ground truth, and we told the ground truth which labels we care about. So now we have to do the same thing for validation. And it is essentially identical, except now we pass it a different array. So instead of PER01 100, I'm passing it PER01 120. And so that's essentially how you would configure your data in SR. This example is a little, this would be a little bit annoying to do for this full data set. So if we go back over here, I've configured a whole bunch of different samples. This is easier to do since these are 2D and fairly small data sets. It's a little bit annoying to do. Ideally, you usually just have like one big raw volume that you can pull from. Like in the case of the liver, it was just a massive raw data set, and then a couple crops that are 3D and thus contain a lot more data. So it's easier when you don't have so many data splits, but it can be done with a lot. In this case, I used a Python script. So there is a Python backend for DeCapo that makes it that is quite easy to use, and will let you configure stuff there as well. So if you're familiar and comfortable with Python, you can do that. We're working on tools to make this front end easier to use and just faster and more convenient to configure lots of data. So that is the epithelial data split. Next, we might want to do a custom architecture. So this is another fairly quick one that we can just do right here. Gonna call it epithelial 2D unit. Add an input shape. So I don't know, pick something somewhat reasonable. And then here are parameters that are somewhat specific to units. So this is something that I might expect someone from like Jan's lab or Stefan Salford's lab or the Serini lab, the Suraga lab, to configure. Someone who's familiar with units and familiar with machine learning might make some units and test it on their data sets, maybe a toy data set or two, maybe some actual projects for someone, and then other people could reuse the data set. So I don't really expect the average user to know everything in here, but in case you are familiar, you can configure it. So a unit, the number of feature maps at the very top level, I'm going to put 20 in here. I can pick large numbers here because this is 2D, so it won't take up quite as much memory. Feature maps in is just going to be 1 because we're taking raw data with just a single channel. Number of feature maps. This is the number of feature maps at the top level of the unit. The feature maps out is the number of feature maps produced by your unit. So they're slightly different, but similar. In this case, I'm just going to keep it at 20. And then fmap, increment factor. Every time you downsample in a unit, you multiply the number of feature maps by some number to keep to avoid too much of the information bottleneck. So that's where you get down to the bottom level of the unit. You still want to be able to represent a lot of the data from your input. So that I'm putting 10. For downsample factors is how much you want to downsample at each layer. So I'm going to downsample by 2 by 2 on the first layer and 2 by 2 on the second layer. So it's just a 3-layer unit. At the top, we should have 20 feature maps, then 200, then 2000. Kernel size down is optional, kernel size up is optional. These are all optional. Upsample factors doesn't say optional, but it is optional. That is, this unit supports outputting labels at a higher resolution then the ground truth is input. So for example, if you want, in the liver data set I was showing earlier, the ground truth was at 8 nanometers and the output was at 4 nanometers. That's nice for objects that are particularly small, and you might want to have a couple more pixels there so you can define the boundary a little better. Constant upsampling is when you do the upsampling layer, whether you want to have that be learned or if you just want to naively copy values so that they're to upsample. And then padding, your options are essentially just valid or same. So valid means you will avoid filling in zeros at the boundary to keep the convolutions the same size. And same means you'll pad with zeros so that the output is the same size as the input. And then we can save that. So that's the task data split architecture. We can also do the trainer. This is fairly straightforward as well. We give it a name, trainer epithelial. We give it a batch size. I'm just going to do 1, learning rate, put in something reasonable, number of data fetches. DeCapo will automatically start however many workers you want on a CPU to fetch data for your GPU in a separate thread. And then augmentations, this is stuff like if you want to add a little bit of like this, a little bit of gamma noise. So like between 0 and 2, for example. And then the snapshot interval, how often you want to save snapshots. This is optional because it's nice for debugging, but if everything's working fine, then you don't really need it. So save that. So, okay, let's pick these options. Click it. So it's now write whatever configurations you select. That will then provide a run that combines all of them. And you can pick, for example, multiple different trainers, and it will give you the combination of all possible options. So if these units are slightly different, you can use both of them with all different learning rates, select them all, and then hit train. So I'm just going to start one of them. This one. Let's do 3 repetitions, 10,000 iterations, validate every 2,000. And if we submit and go over to monitor runs, we should see some of that data slowly trickling in. So let's select here. So some of these I've already run in advance. So this one, for example, I've run in advance, and you can see the validation score is going up, the loss is going down, and we can load in some of the snapshots. So again, this is noisy. This is definitely an aspect of DeCapo that we are working on, trying to make tools so that it's a little easier to load in just the data you want to see rather than all the data that is available. So in this case, I've gone ahead and preloaded a specific snapshot and validation block. This might be the last thing we look at today because we're getting close to the end of the meeting. So on the left is a snapshot. We can see that the raw data that was passed in right here was cropped from, looks like it was cropped from the boundary of one of the training examples. but you can see the cells and you can see the membranes. And you can see here I've loaded in the prediction. So it seems like this is after 95,000 iterations. So it's starting to predict the membranes quite nicely. Right there. Brighten that up a little bit. So it looks like it's doing pretty well on the membranes. There's a little bit of noise around the boundary, and that's because if you look at the weight applied to this prediction, the weight mask is in here. You can see that it's 0 sort of near the boundary here. This is because we don't know where the membrane would be if we continued it out here. So I mentioned this before when we were creating the task, but there's a little check mark for masking out parts of the volume where we don't know the answer. And this is it, essentially. Right? We don't know where the membrane might be. It could be sort of 1 pixel from the boundary here. And if it were, then all of our distances defined in here would be wrong, because the distance to the nearest object right on the boundary here would be almost 0, rather than whatever distance it is to the nearest actual ground truth that we see. So during training, we mask out these little parts on the boundary that we don't know the answer to. And you can see that defined on the weight, and then you can also see sort of what happens because of that during the prediction. We don't really force the network to predict anything specific there. We don't care what it does, so it does something that is probably a little bit unreasonable and annoying, but that can be masked out. Since it's really only on the boundary. Everywhere else, you're going to get decent results. So that's the snapshot. On the right, we see the validation volume. I don't know why this is over here. Don't need it. So here, this is the raw input. The ground truth membranes are these sort of single pixel membranes, and then our best membrane prediction is right here. So it looks like our model is predicting slightly wider membranes than it needs to, but for the most part, it's doing pretty well and following the ground truth. We can see some differences if we flip back and forth. Like over here, it looks like there's a gap. Down here, where the membranes get closer, there are some gaps. A little loop here, the ground truth sort of goes around, our predictions go straight. Yeah, that's the little toy example, and it's right near the end of the meeting. So I think I'll stop there.
Thanks very much, Will, for walking us through the tool again. I have one question to start with. If I were, say, a postdoc in one of the labs and I had collected some data and I wanted to do a deep learning project on the data, but wasn't super comfortable with Python or hadn't done a deep learning project before, I'm assuming that's kind of your target user base. Give me like a short summary of the first 5 steps to learning the tool. I mean, 5 is an arbitrary number. I just mean like the first few things very practically. What website do I go to? What do I download? Who do I email? Just like if you can, concise instruction sort of format.
Okay, good points. I probably stopped a little earlier than I should have. But here we go. So how to get started. Currently, there are about 5 steps, and they are somewhat involved. So this is an active area that David is helping me out with. to improve. But for now, if you wanted to get started, there are some GitHub repos that you need to clone. I can give you specific links to those if you're interested. You need to create your own Conda environment, install the dependencies, manage that sort of stuff. You need to start your own MongoDB server. Your own MongoDB is where the training stats, like your loss and your validation scores and all that, are stored. Or your configs are stored up there. So you're going to need a MongoDB if you want to run this. The file server is nice to have because that's what lets you click on these links and open your accounts of views. So again, very nice to have. You're probably going to want it. Your experience on DeCapo will probably suffer a bit if you don't use it, but you definitely can. If all you want is to run something, you can ignore the file server. And then also you're going to want to run the DeCapo server on a machine with dsub access. So if that's like a workstation you have up in Janelia on the internet, that's fine. Basically, anywhere with dsub access, and it doesn't really need to be a particularly strong server. All it's really doing is serving this front end to you and submitting jobs to dsub. It's doing very little processing.
So by my guess, I would say that the average postdoc who's done some data processing as part of their PhD is going to be very comfortable with steps 1 and 2 if they're from the Python universe. And even if they're not, those are things you could learn in half an hour, really. But steps 3, 4, and 5 are probably going to be pretty unfamiliar to them.
Yeah.
So I'm not sure to what extent your shop is open for customers yet, so to speak. But in place of steps 3, 4, and 5, would you say that an appropriate thing to do would be just email Will and that he might have an hour or two to Zoom with you to help set those steps up? Or would you say you're not quite ready for that level of interaction yet?
I would say if you're really interested in getting started, feel free to email me, and yes, I could help you get all this set up.
Okay, because those things to me, they don't seem like particularly hard things to do that would take time. You just have to know how to do them. And that in an hour-long Zoom, you could just share screen, do it for the person who wants to run DeCapo, and then they're all set to actually use the GUI part of the tool, which is more or less self-documenting, and the rest of your presentation teaches how to use it. But exactly. I would say that these steps 3, 4, and 5 are going to be the stumbling blocks for most new users at the start of things.
The only caveat I would say here is that I don't really recommend this route because DeCapo is still being developed. There's still changes happening. So you will, if you go this route, you'll be responsible for you've got to update your GitHub packages and all that as DeCapo is updating. That might be a little harder to manage. If anything about the MongoDB storage changes, it's quite annoying to manage database state. So if you wait a little bit, coming soon, this is what David is helping me out with a lot, is getting DeCapo set up with Nextflow Tower. And Nextflow Tower is a service that will let you submit jobs to like a bsub cluster through the web, which would be awesome because then we can have a single MongoDB, we can have a single file server, a single DeCapo server that everybody could just log into. And then getting started would be just 2 steps. You have to go to the Nextflow Tower, create an account, and then configure cluster login credentials. So your username, SSH key. I've got it set up on there, but I think that's really just about it. Those 2, and then you have to store your Nextflow Tower API key with DeCapo, and then that should be it. You can log into DeCapo. You could see everybody else's configs, which would be a big advantage over doing it your own route, is you won't be sharing anything with anyone else. So if like I create a nice UNet that you want to use, you wouldn't be able to use it on your copy. But if you wait until we've got a setup that can handle multiple users, you'll be able to see my configs. I can create a UNet for you, just put it up there, and then you could take it, use it on your data, use a trainer that I configured to have reasonable augmentations, and then just run with it. So
Okay, that makes a
think-
lot of sense. So the advice to somebody in the situation I described before is, if you're really eager to start right away, you can spin up your own stuff like this, which means learning how to set up a MongoDB server, a file server yourself, or asking for help and seeing if you can get a meeting to get things set up together with one of the developers, or to wait a little while and like if you have a little bit more time, to wait for this more convenient way of accessing all the tools to be set up.
Exactly. This will be significantly more convenient once this is set up. So if you can wait just like a few weeks, I would say please do. But if you absolutely need to get running now, you want something as soon as possible, I guess email me,
Cool.
and I'll see what I can do to help you.
And I'm assuming that nothing about how the usability of the tool changes based on the access method like this. So everything you showed up until this point with setting up models, setting up data splits, running augmentations, things like that, that's all constant, regardless of whether you spin up your own servers or access it through the Next4 tower.
Yeah, that's all constant. Yeah, the only difference would be sharing. So right, if you set it up now on your own, you have to configure everything on yourself. If you set up later, there will probably be architectures to choose from. There'll probably be data sets to choose from, learning trainers and tasks, or and then you just configure whichever new component you need. If you have a new data set but you want to reuse existing task architecture trainer, fine. If you have a previous, if results have already been, like if jobs have already been run on a specific data set, but you just want to try a new architecture, then you would just need to configure a new architecture. So yeah, all else stays the same. The only difference will be the ability to share configs with other users.
Yeah, which is a huge advantage. I think for platforms like this, that's the key feature that's necessary, is being able to utilize work that other people have done within the platform itself. Yeah, that's awesome. Okay, I think there were, like, Jody, you had a hand up, maybe Conrad did too. I'm not sure.
I can start. So it looks good. And I wanted to ask about your current setup and someone having to download repos and start up servers and do all that. Have you guys considered just putting it all into Docker containers with a Docker Compose file, and you could just start the service up? It would pull the containers from Docker Hub and it'd be like a one line, got a service running.
I've definitely considered that. Conrad recommended that as well, that we should containerize this, just because it'd make it easier for Nextflow as well. Yes,
Okay.
I just have no experience with Docker. I think I've touched like 2 lines of Docker in my life.
Sure.
And
So
so
just to clarify, Will, I think what Jody's suggesting is Dockerizing the services, not the things that you're running, right? Not the machine learning.
Oh yeah, I'm
piece.
talking about the HTTP server and the MongoDB and all that. Yeah. So that someone who wanted to start up the service at their own institute or whatever could just Docker Compose up, and it would grab all the containers and start the service for them, and they just have to log in.
that's definitely something I want to do. Jan's mentioned Singularity as well, which I understand to be essentially the same thing. Yeah, I just have very little experience with that, so I need to take the time to do some reading and get that set up and running, and provide the images. Yes.
Okay, cool. And then the other
Exactly
question
what-
was, so this is it's relying on the fact that you can B-sub these jobs to a cluster. So if someone wanted to use it on a cluster that wasn't using B-sub, is it fairly modular to change those commands out?
Okay, so
Or we're not really worried about that yet?
Well, so this is one of the very nice things about Nextflow Tower. Having more so supporting more users is one big part of it. Another big part of it is that Nextflow Tower supports sort of arbitrary run environments. So you'd be able to configure a different cluster if you want. You'd be able to configure running in the cloud if you want. That will break things such as the file server, right? If you point your data to files that exist on Amazon, but your file server is running on the Janelia file system, it's not going to load any of it. Yeah, that is also a very exciting aspect of Nextflow Tower that'd be nice to use.
Cool, thanks.
So my question was you're pushing this obviously in a direction of like user friendliness and eliminating the coding aspect. I wonder, have you thought about also supporting like notebook users who might want to customize some of the code that's running and then but still take advantage of what you're offering in terms of the the hyperparameter optimization piece and all this?
Yeah, so the HTTP server that you see is it's a completely separate component that just works with the main DeCapo backend, which is all in Python. So it's very easy to work with DeCapo in Python. For me, I'd even say it's easier to work directly from Python than from the front end, just because I'm familiar with Python. I can script, I can load in, like if I want to configure a data set with 500 training volumes, I don't want to like sit on the front end and like add each of those manually if I can write a quick Python script that leads through however many folders. So that's definitely doable. It's very nice to use from Python itself. I have not tested it significantly from a like notebook, but as far as I'm aware, there shouldn't be any conflicts between the notebook server and anything DeCapo does. So I think it should be fine.
Cool.
So I have like 1.5 questions related to that, and then also another one that just occurred to me. So you store pre-existing architectures for training in DeCapo, but do you also have a place to store pre-trained models? To say, for example, I want to use a UNet, but not just a randomly initialized UNet, but this one that someone in a different lab already trained a lot. Or you could, for example, grab some open source available architectures that have already been trained. Like you can download the CellPose model, for example, and put it in there and say, here's an alternative way to train CellPose where we'll handle a lot of data splits and augmentations for you, for example. But if you want to update it with some of your own training data, you can do it in DeCapo. There's got to be other neural networks too that are just open source available that you could add.
Mm-hmm. Yeah, so okay, a couple things about this. First is that on the back end, if you're working from Python, this is already somewhat easy and convenient. You can, if a previous DeCapo run has been done, someone trained a model on DeCapo for, I don't know, like 2 million iterations. And now you don't want to redo that. You just want to take their model and fine tune it on your data set. That's doable. As long as you use the same architecture and you use the same predictor, so the same task, essentially, because that does modify the network a little bit to define the number of output channels, right? So as long as you use the same architecture and task, then you're good to go. You can change the data split. You can change the trainer. So if you want to, you don't want to do so many augmentations during fine tuning, for example, Perfectly okay.
Just to clarify that how that exactly works, you would, instead of saying like just train a unit that's randomly initialized, you would provide the initialization weights. So from DeCapo's perspective, it says I'm just training a unit like it would at any other point, but instead of randomly initializing my weights, the user provided a known set, and you happen to know that those weights came from a previous DeCapo run, but DeCapo doesn't necessarily keep track of that.
Well, so a little nicer, you point it to the DeCapo run that you want to continue,
Okay.
and you tell it which criteria you want to use to select the best model. So this is something I didn't really talk about during the presentation, but DeCapo has a fairly fleshed out concept of criteria. So you're validating on some volume, maybe mitochondria, and you get the Jaccard score, you get variation of information, you get false positive rate, false negative rate, whatever you want. All of these can be optimized. And so there are going to be very many different best models depending what criteria you optimize. And so you point to the run you want to continue, and you tell it which criteria you care most about, and it will automatically fetch the weights from that run for the best possible model according to that criteria, and we'll continue from it.
Okay, and that is stored in the MongoDB server?
Yeah.
The result of that run and the network itself, like the train network is in there, all the weights?
No, the weights are stored on the file system.
Okay,
They're
okay.
stored locally, but
all the metadata about like when it was run and what criteria was selected and everything, the configuration of the DeCapo run is in the MongoDB server. So once you've got this all running in Nextflow Tower and users can share, I can figure out that like person in other lab ran this instance on this architecture on this data and say, oh, that's a good candidate for transfer learning to my data. So I'm going to try training that instead of training something brand new.
Exactly. Yeah. And that is very nice and convenient. It's not supported on the front end yet. So there's no option for telling when you create a new run, there's no sort of like text field input to say like which run do you want to continue off of and what criteria do you want to pick.
Yeah. That was my 1.5, one of my 1.5 questions after Conrad's comment is that I do think you'll have just as many users who are like competent in notebooks as who want to use the front end. Like I think that anyone who is doing some MATLAB or Python during their PhD will probably prefer notebooks like you do in the long run. And then some people who are like, I've heard of machine learning but never done any programming will want the front end. So I just to bump his comment, it definitely makes sense to support both, I think.
Yeah, I think I hope so. I think that'll be cool. It'll be cool getting like a large variety of people interested in using it. Yeah, I'm very excited about this Nextflow Tower thing, getting that started.